Thank you again. So as promised in the title, I'm going to start with a story from under the poisonous umbrella. So let's move back in time to 1978 to London. This is the time when a prominent Bulgarian novelist, Grigory Markov, is walking down the Waterloo Bridge over the River Thames. You can see Markov along with his family on the picture in the top left, and the Waterloo Bridge looking down towards Westminster Abbey on the right. Markov is 49 at this time. 9 years earlier, he deflected along with his family from his home country, Bulgaria, to the UK. The Bulgarian communist government started banning his plays and novels, so he could no longer support his family. So he's walking down Waterloo Bridge towards the bus stop shown here on the slide. When he finally gets to this bus stop and the bus arrives, he feels a sharp pinch in his right thigh. Markov looks down, but doesn't see anything suspicious, so simply ignores the whole incident. By the time he gets home, his right thigh is swollen and painful. He talks to his wife and recalls a man holding an umbrella who was standing very close, unusually close to him at this bus stop. Within the next couple of days, other symptoms kick in. He now has severe nausea, has problems swallowing and breathing, and he vomits with blood. So he goes to the hospital. In the hospital, his condition worsens very fast, such that at the day 4 after the incident, Grigory Markov dies. Obviously, his entire family and wives are shocked and in disbelief and want to know what happened. So they ask for autopsy. During the autopsy, pathologist reveals and recovers a small pellet from Grigory Markov's right thigh, shown here on this picture. It's a really tiny platinum ball with a small diameter similar to the head pin. What's interesting is that the small ball contains holes drilled in it, and these holes contain the poisonous toxin ricin, which killed Markov. So what's the aftermath here? Obviously, Grigory Markov died. We don't really know what exactly happened, who committed this murder, or what was the murder weapon. One hypothesis is that an umbrella using a gun-like mechanism was used to inject this poisonous pellet into Markov's thigh. So it does sound like a James Bond-like Cold War plot here, which unfortunately ended very badly for Markov. Since 1978, a number of documents have been released, and some of them implicate the former head of the Bulgarian Communist Party and other top officials in the Bulgarian government and KGB in this murder, but again, no former charges were ever made. So there are a lot of unknowns in this story. However, the unknown we are particularly interested in are the details of the mechanism which ricin-like toxins use, and in this particular case, so swiftly killed Grigory Markov. Ricin is closely related to Shiga toxin 2. The structures of these two toxins are shown on the slide here. They both are small, approximately 70 kilodalton proteins composed of two subunits, A and B, linked together by a disulfide bridge. In case of the STX2 toxin, the B subunit contains 5 identical parts, whereas the B subunit of ricin is monomeric. Ricin can be extracted from castor beans, whereas Shiga toxin 2 is produced by some strains of pathogenic bacteria, including Shigella dysentery and some strains of E. coli. In case of STX2, the B subunit binds to GB3 receptor on the surface of target cells, and it's internalized by the clathrin-mediated endocytosis. In the Golgi apparatus, the A subunit is proteolytically cleaved into A1 and A2 fragments. A2 fragment remains bound to the B subunit, whereas A1 and 2 and A2 fragments, as I mentioned, are linked together by the disulfide bridge. This disulfide bridge has to be reduced in order for the toxin to be active. Now, this both toxin work by binding to the ribosome and shutting down protein synthesis. Reduction of this disulfide bridge takes place in the endoplasmic reticulum. As the result, A1 subunit is released, which then travels to cytoplasm and binds to ribosomes. The A1 subunit of STX2 binds to the ribosomal stalk shown on this picture. Ribosomal stalk is a highly flexible structure extending approximately 15-20 nanometers beyond the ribosome. So as you can appreciate, this is a nightmare for structural biologists. In eukaryotes, the stalk is composed of 5 proteins. The base of the stalk is formed by the PO protein, to which 2 P1, P2 heterodimers bind. All P proteins contain C-terminal domains shown here in red, and these C-terminal domains are responsible for binding to STX2 and other toxins like ricin. Once bound, the toxin is loaded onto the ribosome, and specifically it binds to the so-called sarsin-ricin loop located in the 28S ribosomal RNA of the large ribosomal subunit. The A1 subunit of STX2 has catalytic activity of N-glycosidase. Specifically, it cleaves adenine at the position of 2660 of the SRL loop. As the result of the depurination, the conformation of the SRL loop changes. So the real job of the stalk of the ribosomes and this SRL loop is loading of the elongation factors required for protein synthesis. Elongation factors such as EF2 or EF2 alpha, which are counterparts of bacterial EFG. As the result of this conformational change within the SRL loop, this translation factors cannot bind to the ribosome such that are active. As the result of this modification, protein synthesis is shut down. So this was the introduction to the project, which is very much live and ongoing. The immediate goal of this project is to determine a structure of the STS in complex with the ribosome, to understand the mechanism of this interaction. The ultimate goal is to use these structures for rational drug design and optimization of fragment and peptide inhibitors blocking this interaction. This project is conducted in collaboration of my lab and the laboratory of Nilgun Toomer from the Department of Plant Biology at Rutgers. Scipion Lin from Nilgun's lab carefully investigated the interaction between STX2 and ribosomes and measured the affinity and the KD of this for this interaction to be 3 nanomolar. Scipion also provided us with the samples of the yeast ribosomes and STX2. What we did, we mixed these 2 proteins in different stoichiometries, ranging from 1 to 1 to 1 to 10, optimized grid freezing conditions, and determined structure of the ribosome. So this slide shows some steps from this analysis. You see a representative micrograph, some 2D classes, and 3D structure. I should have mentioned that what we are hoping here was that we'll be able to trap STX2 while being bound to the SRL loop on the ribosome. So it was a very ambitious project. Unfortunately, the structure does not reveal the densities that would correspond to STX2. So please zoom in into this inset at the bottom of the slide. You see here local resolution mapped on the surface of the large ribosomal subunit in the region where the ribosomal stalk is located. So overall structure in this region is high, reaching beyond below 3 angstroms in many regions. However, this is not the case for the bacterial ribosomal stalk, which indicates high flexibility in this region. We try to employ focus alignments with or without density subtraction to visualize STX binding to the ribosomal stalk. However, this analysis again did not reveal density indicating STX2. Well, it was a long shot anyway. So we need to modify our strategy. So before I move on and describe this new strategy, I would like to briefly show you a processing pipeline which we use to calculate the high resolution structure of the ribosome. We calculated this structure. Obviously, I don't want to go into details here. We calculated this structure using a program Scipion, which in fact is a Python platform integrating various algorithms of different cryo-EM programs like Relion, CryoSpark, Eman2, or other versions of Eman, and so forth. So pretty much you name it, and probably Scipion contains this algorithm. So it's a very convenient. We love this program actually. So going back to my story, our new strategy involved targeting a structure of the complex between isolated ribosomal stalk in complex with STX2. The sample of the purified ribosomal stalks were provided by a collaborator from the University of Lublin in Poland. So this slide briefly summarizes a single particle analysis workflow, which we use to calculate the structure of the complex. Briefly, approximately 35,000 movies were recorded using Titan Krios instrument in a super resolution mode. The initial stages of image processing were performed in CryoSpark, namely ab initio model building and some initial refinements, which produce a 6.8 angstrom structure. I then transferred atomic coordinates for particles from CryoSpark to Relion using PyEM and in-house scripts. That's probably too many details for you already. And then carried out further refinements in Relion, including Bayesian particle polishing, CTF, and multi-body refinements. Currently, the structure has a 4.3 angstrom resolution. I continue processing it and refining it further in the program Scipion, which I described previously. So let me briefly describe some of the structural features of the structure. Actually trying to move a screen here, which is blocking the right side of my screen. So I cannot really show you what I wanted to. Okay, now it's good. Thank you. Okay, so you see the structure in the middle. At the bottom, there are B subunits and the A1 catalytic A1 subunits on the top. The picture in the top left shows the hinge region where the active site is located. I mean, it is located close to this region. The bottom left shows well-refined densities corresponding to individual monomers building the B subunit. In the top right, you see the density corresponding to the A1 subunit. This part of the structure contains additional density, highlighted here in green, which can be fitted with the 11 amino acids from the C terminus of the CTD of one of the P stalk proteins. So the sequence is given here, and it is well known that this 11 amino acid peptide binds strongly to STX2. In addition, the structure reveals the flexible loop connecting A1 and A2 subunits, which is not visible in the crystal structure. While they were working on this project, meaning cryo structure determination of the complex, our collaborator determine the crystal structure of the STX2 with P11 shown here. So firstly, the binding pocket identified in the crystal and the cryo-EM structures is the same. This is the same region on the surface of A1. Now, secondly, in the crystal, only 6 out of the 11 C-terminal amino acids are visible. Now, thirdly, this polypeptide is unstructured. That's not the correct term here, but it does not form, for instance, alpha helical structure. In contrast, and this slide shows the P11 STX2 binding surface from the cryo-EM structure. The density corresponding to the C-terminal part of the CTD can be fitted with the alpha helix. Now, this slide also highlights some of the conserved residues which were identified in the lab of Nilgun tumor as necessary for the binding. So namely, arginines 172, 76, and 179 are absolutely required for the binding between P11 and STX2. Substitution of these residues abolishes binding. So, as I mentioned, this binding pocket is very similar to the one identified in the crystal structure. However, there are some differences. Obviously, one might contribute these differences to differences in resolutions. We are currently refining the structure to higher resolution. However, you may appreciate that it's not an easy task, given the size of the STX2 is 72 kilodaltons, and we are looking at the binding of 11 amino acid peptide. Nevertheless, I think the current resolution already provides a lot of very, very useful information. So by comparing these two structures, we see some residues which have been implicated in the P11 binding from the crystal structure. However, in the cryo-EM structure, they don't seem to be in the hydrogen bonding distances with P11. And vice versa, there are some residues, like the one highlighted, for instance, in yellow. They seem to be involved in the interaction with P11 by the cryo-EM structure. However, they were not implicated in binding by the crystal structure. So what's going on? So one interesting observation, which was very recently made by our collaborator, is that once the N-terminal serine is phosphorylated of this peptide, it changes the propensity of this, changes the structure, secondary structure of this polypeptide, and increases in propensity for alpha helix formation. So in the crystal, the polypeptide, which was soaked into crystal.
Time to wrap up, Arek.
Okay, okay, was phosphorylated, whereas in our cryo-EM structure, we have a mixture of the phosphorylated and unphosphorylated peptides. So this could explain the difference. So I want to wrap up by outlining the mechanism which is used by STX2 for inactivation of the ribosome. A1 subunit binds to the CTD domains of the P proteins and is loaded onto SRL loop on the ribosome. The purination of the SRL loop deactivates ribosome, since the P-stock binding and active sites are not overlapping. This binding does not interfere with catalytic activity. After the depurination of the SRL loop, the affinity of the A1 subunit to ribosome decreases and consequently dissociates from the ribosome. And I would like to finish by acknowledging people who contributed to this project. So here you see pictures of the folks from my lab and friends. And this is a collaboration with a lab of Nilgun tumor, and particularly Xiaoping Li, contributing enormously to this project. We're lucky to obtain R1 funding for this research. I mentioned Przemek Grela, who provided a sample of the ribosomal stock. Carlos is our long-standing collaborator. Data were collected in NY Structural Biology Center and also the Rutgers facility supported by Jason Emre. I would also like to thank Rajesh Patel, who runs the standard EM facility, and Stephen Burley and Ken Dalberg for continuous support of this project in our lab. Thank you very much for your attention.